Okay. Hi, everyone. Welcome to this webinar. I'm Mingkun Zhang at GemPharmatech, and I'm very happy to have the opportunity to share with you about the NeoMab platform at GemPharmatech. This is the content I will introduce with you in this webinar. When we look at the drug approval history, there are more and more fully human antibodies are developed and approved, especially in recent years. It was worth noting that a significant majority, more than 2/3 of them, are derived from transgenic mice. The transgenic mice have become valuable tools in the field of therapeutic antibody discovery. There are essentially three major types of monoantibody discovery technology: the traditional hybridoma approach and some relatively newer technologies such as phage display and single B-cell sorting. Traditionally, when we use the wild-type mice like BABC for immunization, as indicated in here, we could obtain antibodies with the desired properties through hybridoma. However, there was a catch. These antibodies had to be engineered to humanize the mouse amino acids to make them less likely to provoke an immune response in humans. The phage display technology could build large library to screen human antibodies. But there are unnatural pairing, which might be still immunogenic. Single B-cell technology, on the other hand, could also be used to discover antibodies from human PBMC, but it often results in antibodies with lower affinity. Now, when we use the transgenic mice to immunization, all of the antibodies they produced are directly encoded by human genes, and their B cells are selected within their natural endogenous system. These approaches comes with a lot of advantages. First, it saves time and money because there's no need for humanization engineering. It also reduces the risks associated with immunogenicity. Another significant advantage is that these transgenic mice are compatible with all screening technologies available. So, transgenic mice have revolutionized the way we discover and develop therapeutic antibodies, making the process more efficient, cost-effective, and also less risky. To bring the transgenic mice NeoMab model into existence, megabase-sized human fragments were introduced into the mouse genome by chromosome engineering technology. Specifically, we replaced the variable regions of the mouse heavy chain and kappa chain with human genes, while keeping the constant region of both the heavy chain and light chain intact. This strategy ensured the interaction of B-cell FC and FC receptors present on the surface of many immune cells. Now, why is this important? You may ask. Well, it is because the interaction are essential for normal immune cell development and immune response. Preserving them, they ensured that the mice maintain their ability to produce a proper immune response. In order to help the development of different antibody drug modalities, 3 versions of models are designed, including the typical standard IgG for mono antibody development, and also the heavy chain only version and common light chain version for development of the heavy chain only antibody and bispecific antibody development respectively. More importantly, BABC was selected as the genetic background of NeoMab model. In practical terms, BABC stand out for the good humoral immune response, making them preferred choice for antibody discovery research. When we examine their genetic composition, a compelling difference emerges. BABC mice possesses fully functional MHC genes, as you see in this table, setting them apart from other strains where at least one allele often carries defects. Specifically, when we look at the MHC2, a key player closely linked to the humoral immune response, we found that BABC mice possesses 2 functional MHC alleles. All the MHC locus are functional. This genetic advantage implies that BABC mice likely have a more extensive antigen presenting capacity. Here's an example from a research paper published last year. They used the flu peptide vaccine called HANP to immunize 2 groups of mice, as you can see here, a BABC group and a Black 6 group. The BABC mice have fully functional MHC IA and IE molecules, and they respond well against the immunization. On the other hand, the B6 mice lack functional IE, and they didn't show a response to the immunization. To assess the phenotype and the practical potential of NeoMab mice, it's essential to examine various aspects of the model. This includes analyzing human VGJ gene utilization, assessing B cell development and maturation, and also evaluating the model's capability for antibody production. We did a comprehensive study by conducting deep sequencing of B cell receptors using the PBMC obtained from NeoMab mice, humans, and BABC. In the left figure presented here, we can clearly observe that all the human genes that introduced into the mouse genome are indeed utilized for encoding BCRs in the NeoMab mice. We take IgH, for example, but the result is quite similar to Ig kappa, and furthermore, upon closer examination, it becomes evident that variable genes are utilized at a frequency in NeoMab mice that is quite comparable to that usage in humans. Please look at the germline fragments used at a higher frequency in NeoMab mice here. They're also used at a higher frequency in humans. Now, when we talk about human antibody diversity, it is a crucial factor in the antibody discovery. To gain a holistic understanding of the diversity, we calculate the Simpson's diversity index using deep sequencing data. The results indicate a remarkable high similarity between the recombined heavy chain and light chain sequences in NeoMab mice and humans. As reported in literature, the average length of amino acid in the CDR3H in antibodies varies among different species. Notably, the average length in humans tend to be slightly longer than that observed in the mice. And as demonstrated in this figure, the CDR3H length in NeoMab mice closely mirrors that of humans, but significantly diverges from that of wild-type Bobby mice. As you can see here, almost no sequence detected over 22 amino acid long. This data suggests that the structural characteristics of antibodies produced in NeoMab mice have a higher resemblance to those produced in humans. In addition, it is important to recognize that the way animal acids are used at various positions within the CDR3 region is unique in each species. Here is a specific example. Look at the 106 and 117. When we examine the frequency of amino acid usage preferences at these positions, again, NeoMab mice display a striking similarity to humans, but they differ significantly from their mouse counterparts. And next, let's take a look at the immune cell composition in both the spleen and the blood from NeoMab mice and wild-type Bobby mice. Notably, we didn't find significant differences in the proportions of various types of immune cells between NeoMab mice and Bobby mice. This suggests that their immune systems are competent. Moreover, there doesn't seem to be any obvious changes in the development of B cells after a genetic engineering process. This is evident from the similarity between the B cells at different stages in both groups of mice. In addition, as you can see here, various isotypes, including IgG, IgM, IgA, and IgE, are identified in serum of both groups of mice, indicating that expected class switching has occurred. Now, what does all of this mean in a big picture? Well, these findings support that NeoMab model indeed exhibit the anticipated phenotype, which is particularly significant because this model was purposefully designed for the antibody discovery. However, the performance of the model in antibody discovery applications still needs further testing. A mono antibody discovery project was studied in collaboration with a CRO company specializing in antibody drug development. For this project, we chose the widely recognized target PD-1 to immunize the mice and performed antibody discovery as a test case to validate our platform. Following 3 immunizations with human PD-1 protein, we observed that both NeoMab mice and Bobby mice have similar serum titers, ranging from 100,000 to 1 million. Furthermore, the serum antibodies exhibit the ability to bind to human PD-1 and in its natural conformation, which is expressed on the cell surface. One of the immunized NeoMab mice was selected for hybridoma screening. We not only assessed the binding affinity of antibodies, but also their ability to block PD-1 and PD-L1 signaling. And as demonstrated in this table, antibodies with blocking activity have an affinity ranging from 0.1 to 10 nanomole as determined by Biaco analysis. And among these antibodies, 10 demonstrated superior affinity, and 2 of them significantly surpassing BMK in terms of binding strength. Take 10K for F3 as instance, which displayed a KD value indicating significantly higher affinity than BMK. And importantly, the affinities of these antibodies discovered using a NeoMab platform align closely with those of approved antibodies as reported in scientific literature. These antibodies with PD-1, PD-L1 blocking activity were selected for sequencing in this case. And the result revealed different VH and VL genes were used to encode the antibodies. We obtained in a total of 20 unique sequences from this mouse. And here is another case. This is a set of unique antibodies clones with binding activity. More different VH and VL genes are used to encode these antibodies, indicating a higher level of diversity. In this particular case, we conducted epitope binning experiment of the 14 PD-1 antibodies with blocking activity. These results revealed that these antibodies could divide it to 2 distinct epitope groups. Both of these groups were found to be different from that of Keytruda, indicating novel binding sites. Furthermore, we assessed the functional capabilities of these antibodies. As shown in this slide, their blocking activity is comparable with that of Keytruda. We choose 3 of these clones highlighted in gray here, and these antibodies were selected for production and purification of antibodies for in vivo study. We conducted the in vivo efficacy study using PD-1 humanized mice bearing MC38 tumor. 2 different doses were tested, a high dose of 5 mpk and a low dose of 0.5 mpk. These results are promising overall, as the tested antibodies demonstrated the ability to inhibit tumor growth at levels comparable to Keytruda. Notably, one of the clones, 9K6D11, exhibit an even better tumor growth inhibition index than Keytruda. In fact, we conducted tests to assess immune response using various targets. Here's another example. Notably, we found that overall serum titer was remarkably similar between the NeoMab mice and BABC groups. Specifically, it reached approximately 1 million after 3 to 4 rounds of immunization in both groups. This is the serum titer of another target. The titer is about 1 million, which indicates the immune response is robust. Building upon the NeoMab model, we also assembled a dedicated team of experts in antibody discovery to offer professional antibody discovery services to our customer. Once the target is selected, we can kickstart the project. Take the timeline for a project like TAA as an example. From the target selection to obtaining leads, it typically takes 6 to 8 months. Additionally, we also extend our services to include further in vivo studies, ensuring a comprehensive approach to antibody development. In summary, we have some benefits from the NeoMab platform. Firstly, the high quality. The model was developed in the bovine genetic background, which allows for a robust humoral immune response. Our validation data strongly supports the generation of antibodies with high binding affinity and diversity through the NeoMab model. And secondly, unlike many of our competitors, GemPharmatech operates as a CRO company that only provides resources, technologies, and services to help our clients to develop their drugs without any clinical programs. Thirdly, GemPharmatech provides a comprehensive one-stop solution for our clients. We offer a wide range of animal models and preclinical services that can support in vivo studies. Last but certainly not least, our business model is flexible and customer-friendly. We even offer extra discount if you choose to share your data with us. We are committed to delivering exceptional value and results to our clients. The NeoMab platform is actually a powerful tool that allows us to accelerate our customers' antibody drug development in several ways. Firstly, NeoMab offers a fully human antibody repertoire upon immunization. This translates to reduced costs and time associated with the humanization and further engineering of antibodies. Because of the natural endogenous development and maturation of B cells in these mice, the antibodies they produced may be particularly advantageous for future development stages, such as CMC stage. Secondly, the NeoMab platform seamlessly integrates with other resources and platforms available at GemPharmatech, providing a holistic solution to our customers. For instance, we can leverage target knockout animals from the KOAP bank for target validation, MOA studies, and biomarker investigations. We have over 800 target humanized models, as well as disease models ready for use in preclinical studies of drug candidates. In addition, GPT's team can initiate the development of animal models and also in vivo study systems right at the beginning of the antibody discovery project. This synchronization aligns the timeline of discovery and the in vivo animal study system for the drug, ensuring efficiency throughout the whole process. This integration can save up to 1.5 years for our customers. And okay, this is the end of my my presentation. We can provide multiple type of cooperation, the platform license, and also antibody discovery services. We also select several targets to validate our platform. So if you are willing to license our products, it's okay. And we are all the cooperation are welcomed. Thank you very much. And any questions will be appreciated. Okay, here's a question from our audience. Are there different types of collaboration with NeoMab platform? Yes, basically we have two types of collaboration. The first one is execution study. And in this case, the client provides the entire study design. GemPharmatech team will then carry out the study according to the design. We will have detailed discussions with our client to confirm the specific delivery criteria for each part of the project. And also materials required for this study can either be provided by the client, purchased by our team based on the client specified supplier catalog or lot number, and also can be developed by our team if requested. And if the study involves assays, this assay can also be transferred to us from the client or developed by ourselves. The second type is standard study. Here, the clients provides the target without background information, along with the delivery criteria. And we will take charge of both the investigation and the study design. After the clients approve the study design, our team will proceed with executing. Similar to the first type of study, we will discuss with our clients and confirm the delivery criteria for each part of the project with the client. Also, the materials can be provided by the client, purchased by us, or developed by ourselves. Also, all the functional studies, if needed, can either be transferred to us or developed by our team. You're welcome. Any other questions? Okay. Thanks for attending.